related to this competency 13.1. We had talked earlier about oncogenesis. We had also talked in that context about proto-oncogene activation to oncogene P53 also we had talked about. And the sum and substance of everything was that two types of molecular accidents can cause cancer. Either there is activation of the growth promoting gene or inactivation of the growth inhibiting genes. So these are, as we had said, are tumor suppressor genes, and these are proto-oncogenes which code for pro-mitotic proteins, and the tumor suppressor genes encode for growth inhibiting products. So these two types of molecular accidents can foster cancerous transformation, malignant transformation of a normal cell. So most of the spontaneous cancers arise because of these two molecular accidents. Viruses also they cause, we can say, another type of molecular accident. Isn't it quite amazing that such small particles, virus, nucleoprotein, a small genetic corpus, sometimes we can say, some of them they have got very small genomic material. So how they can induce cancer in the humans, that we are going to see. The answer lies, if not fully, then partially in the fact that not all viruses, but most of them, they insert their genomic material into the host cell DNA. So ये virus ने अपना genomic material, it gets incorporated into the host cell. And there are two types of viruses, DNA viruses, which may directly do so. And then there are RNA viruses called retroviruses. They first, with the help of reverse transcriptase, they synthesize uh, complementary DNA. This picture shows in a better way. Retroviral genome. This is RNA by reverse transcriptase. Complementary DNA is produced, which is inserted into the human or the host cell genome. After it gets well entrenched in the human genome, it produces cancer by two mechanisms. But let us examine first. This is the viral genome, GAG, POL, ENB. These are the normal viral genes which are required for viral replication. But what about this VSERC? It is not at all required for viral replication. So then what is it? It is an oncogene. So with this background, we talk about two mechanisms by which cancerous transformation can be induced by viruses. Till now, we have done that the retroviral complementary DNA has been incorporated into the host cell DNA. After that, the viral oncogene, VSERC, it encodes for viral oncoprotein, of course, and rest is same. It's an oncogene, oncoprotein encoded, and it causes cancerous transformation. That is the first mechanism. Inserted oncogene codes for viral oncoprotein, which fosters malignant transformation. That is number one. Second mechanism, even if the virus does not contain an oncogene, it can cause cancer. How it does so? By inserting next to cellular proto-oncogene. Ye just, in a rough diagram, let me show first. Supposing this is a cellular proto-oncogene. The virus inserts in its DNA in neighborhood of this cellular proto-oncogene. And as we had seen in the previous one of the slides, that the viruses, they have got LTR called long terminal repeat, and LTR contains promoter enhancer sequences, etc. Promoter enhancer sequences, they reside in LTR. So we return to this. This is the viral genome और इसके अंदर promoter होते हैं. These promoters will stimulate proto-oncogene and they get overexpressed. This picture shows in a most explicit way retroviral genome insert हो गया. Under influence of these promoter, the cellular proto-oncogene will be overexpressed. Excessive production of proto-oncogene product, and that is hallmark of cancerous transformation. Two questions which I would like to address, that how come virus contain an oncogene for the human? The answer is that in humans also हमने V सर्क देखा था, प्रोटो ऑन्को एक वायरल ऑन्कोजीन था. Its cellular equivalent called C circ is present in human cells. In other words, a proto-oncogene which is present in the human cells, probably this C circ, this proto-oncogene, how it reached the virus, probably it is hypothesized, but it is well accepted now that it was during a previous infection cycle. This went into the invading virus, it was hijacked and then mutated. So this C circ, which was a proto-oncogene, after mutation, it got converted into an oncogene, that is V circ. So I'm repeating this theory. In 1910, it was proposed by this scientist, and still it is accepted. V circ is a viral oncogene, causes connective tissue tumor in chickens. It codes for a viral oncoprotein, protein kinase, which causes cancerous transformation. And now we have said that this is cellular equivalent of C circ. The postgraduates may note that C circ, SRC, this is a proto-oncogene. It is part of the mitogenic signaling pathway, MAP kinase pathway. So this proto-oncogene hijacked into the virus, mutated, converted into V circ, which is a potent oncogene. So it will cause cancerous transformation. One more point I would like to talk. I talked about promoters till now. इसके अलावा LTR also contains enhancer sequences. What are these? These are also stimulatory sequences like promoter, but with a difference. They can act over long distances. Promoter must be inserted in neighborhood of a cellular proto-oncogene. These enhancers, even if they are 10,000 base pairs away, they can act. So they act over long distances. I'm sorry for that. Number two, अगर आप देखो promoter, they are present on the same strand of DNA where cellular proto-oncogene is present and in the same orientation, 5' to 3'. Enhancers, they can act even if they are present on wrong strand of the DNA and even if they are in the incorrect, opposite orientation. Still, but anyway, promoter or enhancers, we have seen there are two mechanisms. Number one, some viruses, they contain oncogene. I'm writing ONC. Some viruses which lack oncogene, they can cause cancerous transformation by stimulating cellular proto-oncogene. In the first case, The oncogene present in the virus is important. In the second phase, promoter and enhancers present in LTR are more important. Okay. Now, we conclude with the retroviruses. Now we come to DNA viruses. But two rare things I'm going to talk about first. So these are some example of retroviral oncogenes. You can see here, circ we have explained, these ones also. HTLV1, this causes invade CD4 cells, and in humans, it causes leukemia and lymphoma. So I must say that you do not bother much about these viruses. You will be studying in details in microbiology. As per competency in biochemistry, the mechanism is important. Now we come to mechanism of DNA viruses. DNA viruses, they are more common. Retroviral induced cancer transformation is extremely rare. Second important point, unlike retroviruses, they do not habitually integrate into the host cell genome. So this is also extremely rare, the DNA virus getting incorporated into the host cell genome. So then what is the mechanism? Several. First and the foremost, I would like to say, some they cause chronic inflammation. The ones causing hepatocellular carcinoma. इसमें क्या होता है? Chronic stage में, when the inflammation becomes very widespread, so that time oxygen-free radicals are generated, and you know that they are cancer causing. Another thing, they cause with chronic inflammation tissue damage also. So this leads to stimulation of the surviving cell, which start over proliferating, leading to cancer. This one is important. I'm going to talk about it. इसको समझने के लिए just revisit this statement. Inactivation of tumor suppressor genes is more common than formation of oncogene in spontaneous cancer. And what are the tumor suppressor genes? We had studied P53 and also, which was the second one, retinoblastoma. Now, supposing if these two were to be inhibited, अगर इन दोनों को inactivate कर दिया जाए तो क्या होगा? As per this statement, cancerous transformation. This is what human papilloma virus does. It encodes for two proteins. It contains two genes, E6 gene and E7 gene. E6 gene का protein product that binds with, I think, P53, and E7 का protein product that binds with RB. Please check it. But the question here is that the important statement that the protein product of E6 and E7, they inhibit two most important of the tumor suppressor gene. So as per this statement, I have written it, cancerous transformation will occur. So the protein products of E6 and E7, they can be called oncogenes, although they act indirectly by inhibiting the tumor suppressor gene. Hepatitis B, you are already aware of this term. This causes hepatocellular carcinoma. So hepatitis B virus, unlike human papilloma virus, it does not contain an oncogene, indirect oncogene. So it must insert itself at random site into the host cell genome. And rest is same as discussed in case of retrovirus. This is Epstein by, this is, I have written here, Epstein-Barr virus. This causes Burkitt lymphoma. This also causes Hodgkin lymphoma. So need not go into mechanism of each and every virus. I think I have told you two or three. That is enough. And then there is another one also, HHV8, which causes capsule sarcoma. You will learn about them in pathology and microbiology. So, ye first I have written this EBV, Epstein-Barr virus, is important. Kyun? Because this was the first time a human cancer was shown to be induced by viral oncogenesis in humans. और उससे पहले वाला था वो चिकन का था, Rose sarcoma virus we had talked about. So finally means summarizing everything. तो summary अब ये है, rare molecular accidents by retroviruses which cause cancer in two ways. Number one, by insertion of oncogene. Number two, by boosting up transcription of normal cellular proto-oncogene. DNA viruses do not habitually integrate. Chronic inflammation leads to cancer. Human papilloma virus का हमने देखा, anti-oncogene suppressor. Hepatitis B integrates into the DNA of the host cell. So I think that's all. I will return to that first slide. तो हमने oncogenesis अलग अलग तरीके के देख चुके हैं, proto-oncogene, P53, etc. So apoptosis we may discuss some other time. So I may be failing in my duty if I do not thank Dr. Parul Gupta and Dr. Edelbert, who have helped me with the slides and with the preparation of this. Otherwise, people of my age and stage, they are very, very poor. We are paupers, just like DNA, viruses, etc. They are genetic paupers. So we depend on you. Thank you so much, Dr. Parul. Now, last thing I would like to say, HIV virus क्या करता है? That kills the host cell. But ये जब oncogenesis होता है, I talked about genetic paupers. एक HPV virus है that contains very few thousand base pairs. So it is in interest, and for its survival propagation, it requires enzymes, DNA polymerase, helicase, topoisomerase of the host cell. So it is in interest of the virus that the host cell not only survives, but it proliferates also. So virus, it is colonizing, and like any colonizing force, it is seeing its own self-interest by causing cancerous transformation. Thank you so much.